Hi guys, so today we can study vision. So vision is organized under three levels and touch was organized under four levels. So we can say that vision is a little bit faster than touch because less organization, less bureaucracy in the body. So vision is a little bit faster than touch. That's the first conclusion. Cool. Vision, we can study vision under three organization. First is retina, second is thalamus, fourth is occipital visual cortex. So this is cortex, it means it does all the computing. This is thalamus, this is a gateway to your cortex, a relay system, if you may understand it like that. And retina is just bunch of neurons, bunch of nervous tissue. And this, the prime function of retina is to understand light, to capture the light, to collect the light and send it to the thalamus, and then the function of thalamus is to send it to occipital visual cortex. Cool. So retina is majorly made up of 7 type of cells, and 6 are involved in vision, and one of them is supporting cell. One of them is in supporting cast, and 6 of them are the main cells. Cool. Let's study retina. Retina is just nervous tissue. It's right behind eyes here, and it's made up of 7 cells. Yes, let's study 7 cells. Cool. First one, the first cell is rod because they are rod shape. Second is cone because they are cone shape. Bipolar because this is a cell and they have two poles. Ganglion, it's a collection of neurons. Horizontal cell, these are horizontal cells literally, and these are like this horizontal cells, and they connect two cells. I will tell you what. Amacrine cells, they connect bipolar cells to ganglion cells, bipolar to ganglion, so they are present here. Horizontal are present here between rods and cones. And then the last one is Muller, Muller, Muller or Muller, whatever you want to call it. So first of all, let's study let's talk about amacrine. The word amacrine means, A means without, macro means big. So a cell without big axon is called amacrine cell. So this connect bipolar to ganglion here. Horizontal connects rods to cones here. What about Muller? Muller is just a supporting cell. I told you, a supporting cast. They are like glial cells which are present in your CNS, right? And there are two type of glial cells, microglia and macroglia. Micro are for cleaning stuff and macro are for supporting. Cool. So Muller cell, aka retina glial cells. Okay, we know this already. So rods, rod shape, cone shape. Bipolar has two pole, one to receive, one to conduct. Ganglion, they are bunch of neurons. Their output axon make optic nerve, and this optic nerve goes to thalamus, and this thalamus goes to cortex. Okay, and these ganglion cells are responsible for generating action potential, and then this action potential can travel long distances via axons. Okay, so we understand the basic concepts of retina cells. These are 7 retina cells: rods, cone, bipolar, ganglion, horizontal, amacrine. That cells that have no big axon, right? That's the concept. Okay, so how they are arranged? They are arranged like this. This is a structure of a retina, and we have 2 retina. So this is a structure. The source of light is here. We push the light here. The first cells is ganglion, then we have amacrine, then we have bipolar, then we have horizontal. I told you horizontal, they connect. What do they connect? Rods and cones. So let's find rods, let's find cones. These are rods and cones, and I told you horizontal are connecting them. So this is horizontal, and you can see rods and cones are connected via horizontal, this like this. Okay, what about Muller? Muller is the glial one. They are just supporting, they are just for the structure. What about other? Amacrine, they connect bipolar and ganglion. Here are the amacular, this is connection. Cool, now we know. First ganglion, amacrine. The sequence really doesn't matter. I mean, it matters in your real life, but Nobody asks about the sequence, but you should know about the sequence anyway. Cool. Let's go towards photoreception. Photoreception is just the way your cell perceive light. So there are two photoreceptors we have. These are both of them rods and cones. Let's study them. So photoreceptors. Cool. Photoreceptors, there are two type of photoreceptors, rods and cones. Rods have something called as rhodopsin. That's very important. Always remember that rhodopsin is a pigment, while cones have something called as idopsin. Idopsin is a pigment as well. Idopsin helps us to visualize blue and green, okay? And this idopsin is great for colors, by the way. So rods is for night vision and cones is for color vision, day vision. Cool. The rods are less in number, and sorry, the cones are less in number and rods are more in number. Cool. Why is that? Why rods are more in number? Well, in night we need more input from the light, so we need more receptors for light in night, because in night you have less light, right? So you need more number of receptors in the night. So that's why God gave us, the nature gave us more rod receptors to deal with night. But cones are mostly used in day, and in day there is enough light to deal. So that's why nature gave us only 5 million and here it's 100 million. Cool. What about other things? Well, rods are present in peripheral retina and cones are present in center. And the central part of retina where cones are saturated, like this, are called fovea. This is only a cone part and it's called rod free zone because there is no rods there. Cool. What is idopsin? Well, why do we need idopsin? Idopsin is a pigment, right? We need this pigment because it is what, this is the chemical that absorbs the light, okay? If we don't have any sort of pigment, it's not gonna absorb the light and you're not gonna able to see colors. When you have some sort of deficiency in this pigments, you end up with color blindness. Okay, what is it? We need pigment as it absorbs light. Cool. Cones. C stands for center because it's present in center. C stands for color because they give color. And C stands for cone because they are shape of cone and they are crisp, more detailed. Okay, what about the things? They are abundant in peripheral retina, like this. This is a retina, for example, and on this way rods are mostly here, like this, on a periphery, and cones are mostly here in the center, and with this part we call something as fovea. Cool. Fovea is for maximum details, right? Because idopsin is concentrated in fovea. Cool. What else should we know? We should know the concept of lateral inhibition. Well, lateral inhibition is basically the concept says that on the periphery there are cells and on the center there are cells. And we know that the central cells are responsible for details and day vision. So what we can do is inhibit these peripheral or lateral cells so that most of the light is concentrated in the center. That's the concept of lateral inhibition. So some cells like horizontal, what were horizontal? Rods and cones are connected with horizontal. What are amacrine? Amacrine is bipolar cells, and what were ganglion cells. So amacrine is the cell that connect bipolar with ganglion. So both of these cells can be used in this concept. What happens is that these lateral cells are inhibited, okay? And what happens is that when they are inhibited, most of the light is concentrated in the central one, the fovea one, okay? Okay, so inhibits the lateral cells, enhanced output from the central cell, of course. The contrast and the sharpness increases a lot. That's the concept of lateral inhibition. So this is it. Cool. What is it? It's melanoopsin ganglion cells, MGS, melanoopsin ganglion cells. Cool. What is it? Well, there's something called melanoopsin, okay? Melanoopsin. It's a pigment, right? And this pigment can be directly stimulated by light, right? And these cell, melanopsin ganglionic cell, I told you ganglion make the retina, the axons of retina that goes to thalamus, right? So these ganglionic cells, they have something called as melanopsin, right? And this melanopsin has something that is called PACAP, PCAP, PACAP, PACAP. What is it? PACAP is pituitary adenylate cyclase activating polypeptide. So it's a polypeptide. It is a protein, right? It's a protein that is found in melanopsin, right? And this PACAP is found in retina, right? Because these ganglion cells are found in retina, and this ganglion cell is going to make axons, right? Cool. So this PACAP has two functions. The number one function is for the hypothalamus. The number two function is pretectal nucleus. Okay, what is number one function? The number one function is for hypothalamus. What happens is that this PACAP in hypothalamus, right? This PACAP in hypothalamus maintains something called as circadian rhythm, right? So what happens that whenever sunlight goes inside your eye, it goes through these cells, melanopsin cells, and what happens that through this whole pathway, sunlight reaches to this PACAP, okay, and this PACAP is actually via this tract called retinal hypothalamic tract, RHT tract. It activates something called as suprachiasmatic nucleus, okay, and this nucleus is responsible for maintaining your circadian rhythm. What is circadian rhythm? Circadian rhythm is when you sleep for 12 hours and you wake up for 12 hours. The night versus the day. This is your normal circadian rhythm. You should sleep in night, you should wake up in the day. And you can actually make sure that it works nicely. Is how you can make sure. You just have to go out in sun for like 10 to 12 minutes or maybe 20, and get enough light inside, not inside your eyes, basically inside your body. And what happens that this sunlight will actually activate this suprachiasmatic nucleus, and then your 24 circadian rhythm will be activated, right? And you will maintain this circadian rhythm. So this is very important. So sunlight for 10 minutes is very important. This is done via retina hypothalamic tract, RHT tract. Cool. What about other thing? It's called pretectal nucleus. Okay, pretectal means it's a nucleus that is present before tectum. Tectum is a part of your brain. Cool. Now you know there is something called tectal. Well, actually there is something called tectal nucleus, and this is pretectal nucleus because this is just before that. Okay, now you understand the meaning of the name. Cool. So via this pretectal nucleus, there is something called as Edinger-Westphal nucleus, and this nucleus is actually responsible for pupillary light reflex. If you throw some light inside this pupil, you will see the pupil will contract. And why is this happening? I will tell you the whole pathway. What happens is that light goes via pretectal nucleus and via Edinger-Westphal nucleus, and this pupillary light reflex is performed. So remember, if you put light in one eye, what's going to happen is that both of the eyes will show this contraction of pupil, okay? Both left and right, both right and left, okay? Reflex is active. Not just the right eye is active and left eye is slobby. if it's happening, then okay, you're not normal. It has to be both eyes. Okay, cool. Now we know about melanopsin. Melanopsin has two functions. First is circadian, second is the pupillary reflex. Cool. Another topic, phototransduction. What is phototransduction? Well, phototransduction is how the photons, the light is traveling. Well, the light travels via retina, right? Cool. Let's study this whole thing. Cool. Well, phototransduction in short is the story of glutamate. What is glutamate? Glutamate is a neurotransmitter, and this is a chemical substance, chemical bioactive bioavailable peptide. Okay, this is a good definition of glutamate. Glutamate is a bioavailable amino acid, and it is a neurotransmitter, and it is used to activate, it is used to excite, a cell. Okay, so it is a story of glutamate, it's a story of neurotransmitter, and it is a story of balance of light and day. Okay, balance of wait, I wrote it wrong. It is a story of not light, night and day. Okay, well, what happens is that you have rods, right? And rods are made up of rhodopsin, the pigment, and this rhodopsin is made up of opsin plus vitamin A. So if you lack vitamin A, you will have problems, because, and this is how the night blindness actually works. Because when you have deficiency of vitamin A, your rods are not working properly, and then you will end up with not being able to see in the night, because rods are primarily important for night vision, because rods are more in number and you need more receptors for night because you have less light in night. Okay, so contain rhodopsin made up of opsin plus vitamin A. And what is what vitamin A is, right? Vitamin A, it's made up, it's present in your body in two forms. Okay, first is 11 cis retina, and other is all trans retina. Okay, and 11 cis retinal, all trans retinal. Okay, this forms of vitamin A can be changed with the help of light. Okay, so this 11 cis retina is found in night, the dark state, and all trans retinal is found in light state. Okay, so with the help of light you can convert this. So in the night you will have this one, in the day you will have all trans. So 11 p.m. is in the night, that's how you remember the name for the night. With the help of light converts this to all trans retina. I will tell you why we need these two forms. Wait. So this cycle, when light converts this 11 cis retinal to all trans retinal, it's called Walls cycle. Walls was a scientist, and he discovered this thing. So we need light to convert 11 cis to all trans. Cool. So what happens in this story of phototransduction is that in the dark state, when you have this thing 11 cis retina, the normal RMP, the normal potential of your cell, resting membrane potential, the charge of your rods is minus 60. Normally it's minus 70. And how it stays minus 60 is that there is a continuous efflux of potassium out of the cell. Okay, and potassium is positively charged, and what happens is that you will end up with minus 60 millivolt. And what happens is that in the night you have this cells 11 cistern, right? Cool. So a thing called funny current, right? Funny current channel remains open. Cool. How? Well, via something called cyclic GMP. Well, cyclic GMP is a second messenger. Okay, and what is second messenger? Well, you have this cell, and something goes to the receptor that is present on the membrane of the cell, and then this receptor talks to the nucleus of the cell via this second messenger. Okay, so cyclic GMP, CGMP, is the second messenger, and this cyclic messenger, second messenger, activates, opens this funny current channels. Okay, and this funny current channel allows sodium, that is positively charged ions, to get inside, and this influx of positively charged ions result in depolarization. What is depolarization? It means the cell jumps from minus 60 to, maybe a little bit of positive, maybe plus 30 millivolt. Okay, so this is what happens, right? So influx of sodium allows depolarization, And this is the usual story of normal cell. So this is cyclic GMP is very important now. And this cyclic GMP allows depolarization, and once the cell is depolarized, once the cell is positively charged, it will produce action potential, and this will result in release of glutamate. And I told you that glutamate is an excitatory neurotransmitter, excitatory chemical, excitatory substance. Okay, it excites the cell. Okay, so you understand this glutamate release. Okay, now let's talk about light. Now when you open your eyes in the light, right, the day has started, sunlight comes, what happens is that this 11 cistern retinal receives the light, and this converts into all trans. We call this thing Wald cycle. And then this G transducin, actually a lot of people, they call this transducin, just transducin. I like to call this G transducin because it's a subunit. It's present inside the cell, right? So what it does is that G transducin activates phosphodiesterase, which is actually called PDE in short form, activates PDE, and then this PDE degrades. So degrades means deactivate. This one, cyclic GMP, the second messenger. So if your cyclic GMP is working, it's nice. If you have presence of light, which will eventually result in PDE, this will stop this cyclic GMP, right? So if your cyclic GMP stops, the closure of funny current channel will stop. Once the closure of funny channel stops, there will be no entry of sodium. Nothing, nada. No sodium will come in. Once no sodium come in, the continuous state of efflux of potassium ions will keep on going on, right? So cell will start to get negatively charged again, right? So cyclic GMP ended with depolarization, and light eventually ended the cell in hyperpolarization state, negatively charged state, because the potassium was getting out continuously. So that's the concept of hyperpolarization, okay? And then eventually the glutamate release is stopped or decreased because there is no depolarization in the retinal, in the rods of retina, right? And this is all done with vitamin A. If you have less vitamin A, you will have less 11 cis retinal. If you have less vitamin A, you won't be able to see in the night. So this is the whole story of phototransduction. So let's study this a little bit more. Let's try to remember this. Well, dark is for depolarization, which is positively charged, right? And I told you glutamate is an excitatory neurotransmitter. So to remember this, depolarization is positive and glutamate is positive. So depolarization equals to glutamate, and it is done with sodium. What about repolarization? Repolarization is negatively charged. So glutamate levels go down, and via CGMP inhibition, right? In retina, in light, hyperpolar, we know this already, right? We just discussed this. So this was phototransduction, right? Let's study something called amacrine cells and visual pathway. Well, first, what was Wald cycle? The cycle from 11 cis to all trans, and then the cycle repeats, because you know why the cycle repeats. It's called Wald's visual cycle. Cool. What is funny current? Well, it's a current, right? And the definition of this funny current channels is that they are sodium channels, and they are located at retina and SA nodes. So if you notice that funny current channels are responsible for sodium influx. and sodium influx later results in depolarization. So we need funny current channels. We call this hyperpolarization. Hyperpolarization means just changing the polarity of the cell. Hyperpolarization, cyclic nucleotide. Nucleotide is channel. Nucleotide is a channel. Okay, so we call this nucleotide channels, right? We call this HCN channel. HCN, this is very important. And we can, these gates, these channels are opened by cyclic GMP. Cyclic GMP is the second messenger, and it can be stopped using phosphodiesterase, this PDE. Okay, these are found in retina and SC node, the pacemaker of heart. Okay, if you have problems in funny channels, you will have problems with the pacemaker of heart, that is SC node. Okay, what is amacrine? We know A means without, macrine means big axon. So the cells which have less or small axons that connect bipolar with ganglion. So these are amacrine. They are purely depolarization cell. They are purely depolarizing cells of retina. No hyperpolarization, okay, role in lateral inhibition. What this is saying is that these amacrine cells are controlled by acetylcholine, because acetylcholine is secreted when the cell undergoes depolarization. Depolarization is positively charged. Okay, so all of the cells in retina can go dipolar and hyperpolar. They can go positive, they can go negative. But the amacrine cells only go for dipolar, and they are controlled by acetylcholine as well. Okay, and they are present in lateral inhibition. Two cells are present in lateral inhibition, horizontal and amacrine. So this is very important. Cool, we understand it now. Let's try to study visual pathway. I have cool explanation here. This looks so complex, but it's not complex at all. Visual pathway. Well, the starting. This is your eyeball. There are two fibers that are coming. First fiber. There are two type of fibers that are coming from your brain into your eyes. Okay. There are nasal fibers, and then there are temporal fibers. Nasal fibers are like this towards your nose, and temporal fibers are towards your ears. This is your ear, right? So this is temporal fibers. Okay, so the thing is that temporal fibers see nasal field, the field that your nasal fibers are supposed to see, and your nasal fibers see your temporal field. Okay, the temporal watch for nasal field and nasal watch for temporal field. Now, why God has done that? Why God has done that? Nasal fibers see temporal sees nasal. Why God has done that? Because our lens in our body is convex, right? So whatever the light is coming inside your lens, it gets inverted. Left looks like left image goes on right, the right side of the image goes on left, top goes bottom, bottom goes top, right? So that's why God was like, okay, if the nature was like, okay, if the lens is, convex, let's interchange your fibers position so that the image we get inside our brain is the right image, right? You don't want to understand the right sign to be a left sign and left sign to be right? You want correct thing, right? You want a good alignment. So this is why your nasal fibers look for temporal field and temporal fiber look for nasal field. Okay, so we know this. Cool. What else is that this nasal fibers, upon coming here, this point is called optic chiasma, whatever you like to call. This is the point where your nasal fiber, they cross, right? And only remember, nasal fibers are crossing, okay? Your temporal fibers don't cross. They go ipsilaterally, like this. But your nasal fibers go like this. Now, why God has done that? Why nature has done that? Well, crossing ensures that both right and left visual cortex get a complete picture of your right and left field, which is essential for depth and perception and stereopsis. What is stereopsis? When you're able to understand that something has 3 dimension, we call that stereopsis. Okay, so that's why your nasal fibers are crossing and your temporal are not. Okay, so what happens is that once your nasal fibers have crossed this chiasma and crossed, the optic tract is formed. Now this optic tract is inside the central nervous system. We call this a tract. This optic nerve, it's outside, and this is the reason why they are named as nerve and tract, because nerve is present in PNS, peripheral nervous system, and tract is present in central nervous system. Nerve is basically a bundle of axon. That's it. Okay, so we now know that optic tract is formed where temporal fibers are ipsilateral. Ipsilateral means on the same side, and nasal fibers are crossing. They are called contralateral. Okay, so after this, these tract, they reach at this point. This is one nucleus. Okay, this is one nucleus. Here is another nucleus present. Okay, and this nucleus, the name of this nucleus is called lateral genticulate body. The word genticulate means knee, because this anatomically looks like a knee, because the shape is knee shape. So they named it lateral genticulate body. Well, if it's named lateral genticulate body, it means there must be something called medial genticulate body, right? It is there. We will come across this. Cool. So this optical tract reaches to LGB. Okay. After this is a relay station. This is a gateway to your cortex. Once your tract is inside this thalamus nucleus called LGB, lateral genticulate body, what happens is that this nucleus produces something called as optic radiation, and then this radiation eventually touches your visual cortex, right? And later on you can understand how your visual cortex works. I will come to this. Cool. So now significance. The significance is that if there is any sort of visual defect, it will depend upon the location of the lesion, right? If there is some lesion here or here, so if there is lesion here, it means what does it mean? It means if the right eye has a lesion, the temporal fibers of the right eye is going to the right cortex. So there will be some problem in the right, eye. I mean, the right cortex will be affected. But if the nasal fibers of right eye before crossing is affected, so the effect will shown in the left cortex, because the nasal fibers, they cross at the chiasma. This is the concept. We understand this concept now. Okay. What else? Thalamus. I told you, thalamic nucleus, lateral genticulate body, lateral knee nucleus, because genticulate is the shape of a knee. And this lateral genticulate body has 6 layers. These are the 6 layers. 1, 2, 3, 4, 5, 6. Okay. What are these layers? Well, the first two layers are in one group, and rest of the 4 layers are in another group. Okay, why? why these two layers are in one group? Because they have one type of cell, and these four layers are in different because they have different type of cell. The first two layers have something called as M cell. M cell stands for big cells because they are big in size, and these cells are responsible for movement because M stands for movement, M cells for M cells. We call this pathway, this M cells make what pathway? They called as magno means big, cellular means cell, pathway, magnocellular pathway. And they are responsible for movement. What movement? The movement of your eye. The movement of your eye, we call this extraocular movement, and plus they can detect any sort of light flickering that is happening. Okay, so they are M cells. M stands for mega or magno, and they stands for movement. So what is termination? Termination is they layer 4 of visual, they end up on where? They end up on layer 4. Both of the group, this group and this group, they both end up at layer 4 of cortex. Okay, what about this one? The other group, the other 4 layers. Well, the other 4 layers are called parvocellular pathway. Well, parvo means small. So they are small in size. We call them P cells. I usually used to remember this parvo with P, and P stands for precise picture. So they are responsible for details and colors. Details and colors. Parvocellular pathway is responsible for colors and details. They also terminate at layer 4 of visual cortex. Layer 3, layer 4, layer 5, layer 6. Layer 3, layer 4, layer 5, layer 6 are parvocellular pathway. P cells. What about other one? The last cell is coniocellular pathway. K cell. They are present between P and M cell. Here. This is M cell. This is P cell. They are present right in between here. We call them K cell. Coniocellular pathway, K cells. They are responsible for the vision of blue and green. Okay, so they are responsible for blue. Cool. Inputs from LGB. LGB, lateral geniculate body, LGB. So input to LGB. What happens is that the input ipsilateral LGB, ipsilateral nucleus receives input from ipsilateral and contralateral eye. What does it mean? It means that this nucleus is getting input from left eye and the right eye, both eye. Because why this is happening? Because the temporal fibers from left eye is innervating this one, right? Because they don't cross. And the right eye nasal fiber innervate this nucleus as well because they cross at chiasma. So which layer receive which fiber? It's a great question. And to remember this thing is that layer, so there are 4 layers, right? Layer 1, layer 2, layer 3, layer 4, layer 6. M cells, P cell. 1, 2, 3, 4, 5, and 6. So what happens is that layer 2, 3, 5 are ipsilaterally innervated. These layer 2, 3, 5, they receive signals from the same side of the eye, right? So what I'm saying is that this is layer 1, layer 2, layer 3, layer 4, layer 5, layer 6. Layer 2, 3, 5 receives communication from the same side ipsilaterally. And layer 1, layer 4 and layer 6 receive communication from the contralateral eye, the opposite side. This is the concept. To remember this, I remember this with 235. It's ipsilateral, right? So U235 is isotope of uranium. I remember this. I don't know why I remember this, but uranium 235 is an isotope. An isotope has an I in it. So I remember that 235 layers are ipsilateral, ipsilaterally innervated. So this is the concept of thalamus. Okay, done. Let's study this thing called visual cortex, how we understand something, right? So this is your visual cortex. In visual cortex, there are two type of cells. Not two type of cells, there are two areas. First one is striated area. Striation means this is a striation, some sort of grooving, we can call it as striation. So visual cortex has one striated area and one not striated. We call that not striated as extra striated. So let's study the striated. Why do we have striated? Well, striated because they have something called as stripes, and these stripes are heavily myelinated. Heavily myelinated means they have a lot of fats on the axons. And if they have a lot of fats, it means they are fast communication. It's very fast. And there's something called as calcarine cortex, which is present in occipital cortex, and the location of this whole visual cortex is in calcarine cortex, which is present in occipital cortex. And this occipital cortex, especially the Brodmann was the name of a German scientist who gave numbers to all the parts of your working cortex. So there are a lot of numbers. There are like 41, 42, there are 25, 20, 28, 17, 18, 19. So what happens is that this, for vision, your Brodmann area 17, 18 and 19 starts working. So area 17 is for raw input, area 19 is for shape detection, and area 19 is for making sense. So 17, 18, 19, very important for understanding vision. And this area, Brodmann, is striped, okay? They have something called a striae of Genesee, also known as striae of Genesee. Okay, what about cells of cortex? Well, we have three types of cells of cortex. First is simple cell and complex cells and the blobs. I will tell you. Blobs make your life colorful. Remember this. And simple and complex cells are used for higher order processing and for feature detection. Okay, they detect minor changes. How they detect? For example, how do you know that something has moved? Well, we know that something has moved because we keep comparing the past of that thing to the present of that thing. We keep that thing comparing. So this is done with these cells. So higher order processing, feature detectors, and this is detect minute changes, right? So this is how we know something has changed or something has moved, with help of simple and complex cells. Both of them, they do it. What about blobs? Blobs are responsible for color vision, and they express something called a cytochrome oxidase. Without this enzyme, you would not be able to see color vision. So it is very important. Cytochrome. Cyto means cell, chrome means color, oxidase means it oxidizes, right? So they express cytochrome oxidase, which is very important. So blobs are color, and they do this thing with the help of cytochrome oxidase.